All right. Hello, everyone. This is Pocket Size Bio, Dr. Casco. In today's video, we're going to continue Chapter 13. We're going to get into Section 3 and Section 4. So this would be Part B of Chapter 3. And let's get to it. Looking to excel in biology? I offer private tutoring, research mentorship, and high-quality study resources designed to help you succeed. You can also support the channel through Pocket Size Bio merchandise or becoming a member. Explore all the links in the description below. Continuing on with Section 3. Section 3 is meiosis reduces the number of chromosome sets from diploid to haploid. So this is the actual process that will enable a cell to reduce the chromosome number from, in our case of humans, it's 46 chromosomes to 23 chromosomes. So we're going to be producing cells that have half the number of chromosomes via this process. So this process in itself has a lot more steps than mitosis, but some of the steps are quite similar. So if you understood the process of mitosis, this one shouldn't be that difficult for you. Okay. Nevertheless, you do see some similarities. One of the main similarities is that meiosis and mitosis, they are still preceded with the duplication of chromosomes, meaning that the DNA is copied during interphase. But in meiosis, there's actually two consecutive cell divisions as opposed to mitosis. And they're broken down into what is called meiosis 1, where you have your first cell division, and then meiosis 2, where you have your second cell division. And within meiosis 1, you have a separation of homologous chromosomes there. And in meiosis 2, you have the separation of the sister chromatids. So you need to make sure that you really understand how to identify the different structures and the different, if I can say, the different modes in which DNA is in, from chromatin to chromosomes to chromatids, the sister chromatids, and then your homologous chromosome sets. So I'm going to show you a little diagram about homologous chromosomes later on. Okay. Ultimately, this process will produce 4 daughter cells, and these daughter cells will have half as many chromosomes as the initial parent cell. As I mentioned before, meiosis 1 is preceded by interphase, where you do have a duplication of chromosomes, okay? And as you enter meiosis 2, there is no duplication. So that's something you need to keep in mind. There is no duplication as you transfer or you transit from meiosis 1 to meiosis 2. DNA does not get duplicated, okay? And sister chromatid concepts and structure you should be familiar with from mitosis there. All right. And then, as I mentioned before, when we talked about mitosis, sister chromatids, they're essentially, I don't want to say melded together, but they're stuck together by cohesion proteins. Okay. And then you have the homologous chromosomes. So this is a new concept where, remember, I've talked about that humans, in our case, we have 46 chromosomes, but these 46 chromosomes can be further analyzed as 23 pairs. Since we get 23 from the father and 23 from the mother, they contain the same genetic information in terms of locus, right? And just the genes that we're referring to. And I'm going to show you a visual on what I mean by that. Okay. So we can have different versions of genes that are referred to as alleles at particular loci, or the locus, the locations in which the genes are found. All right, so what you see in figure 13.7, this is an overview of what we're going to talk about. In terms of interphase, you have this organism here, this single cell that has 2 chromosomes, one from the father, one from the mother. So there's 1 chromosome pair. These are the homologous pairs. Okay. When they get copied, you can still see the presence of the sister chromatids, as I'm showing you here with the cursor. Let's say again, the book shows blue as paternal, red as maternal there. So you have these blue sister chromatids and the red sister chromatids. And then what happens in meiosis 1 is that the homologous pair will separate into each new cell. And then that finishes meiosis 1. And you have meiosis 2 then, where each of these cells will then divide again, where the sister chromatids will be separated into individual chromosomes. Okay? And what we need to see here is that a key point in meiosis 1, that is where you get a haploid number of chromosomes. Okay? So in the beginning, during interphase, you have a diploid cell, and then as you go into meiosis 1 and finish meiosis 1, you will get each haploid cell. So at this point, the cells are now haploid. Okay? Now, just to go further into the stages of meiosis, let's take a look. All right. As I mentioned before, meiosis is broken down into meiosis 1 and 2. And in terms of terminology, each of these subphases have their own number also. So for example, prophase 1, metaphase 1, anaphase 1, and telophase 1. Even cytokinesis could be referred to as cytokinesis 1. So in prophase 1, there are some similarities as we see in mitosis, right? You have the condensation of the chromosomes, right? And the chromatids, they're tightly packed at this point. The spindle, the microfibers, the microtubules, the mitotic spindle is forming. There's a disintegration or fragmentation of the nuclear envelope. So that in itself is consistent. Okay. What is new here is that the homologous pairs will come in and they'll actually pair up at this point. So if you see in the diagram here, you have the paternal and maternal chromosomes, right? So this is a homologous pair and this is a homologous pair. And this big one here, you might think that there are two homologous pairs that are just kind of next to each other, but actually it is just one big one. So in this example that I'm showing you is a cell that is 2n equals 6. So you have a total of 6 chromosomes. So let's count them. 1, 2, 3, 4, 5, and 6. Remember, this is one big one and this is another big one. So there are 6 chromosomes, 3 homologous pairs. All right, so this is one pair, 2 pair, and this big thing is another pair. Now, again, don't get confused with that you have your centromere regions here and they're lining up at the centromere regions. What is happening in this particular case is that you're getting what is called a crossover that I'm going to talk about in a bit. And you can have these events happening at different parts of the chromosome, okay, of the sister chromatids. So let's talk about what that is in itself. A crossover, and this is the main thing that defines meiosis 1 and prophase 1. It's an event that in which the homologous pair, they interchange segments of DNA. So they're crossing over parts of their arms of the chromosomes, and they're exchanging amongst each other. This is referred to as a crossover. Now, more specifically, this occurs during non-sister chromatids. Now, for some reason, the book doesn't show you a diagram, a clear enough diagram on that. So I just got this from Google. Here's the source. I got this from xp.com. And just to give you a representation. So you have your paternal chromosome, chromatids, sister chromatids, and maternal. And what happens is that the arms, one of the legs, the arms of the homologous pair will exchange a piece of their arm, literally it'll break off and then it'll reconnect to the homologous pair. So if you see here, okay, you have the paternal chromatid and then the maternal chromatid. They're going to exchange genetic information. And so you have what are called recombinant chromosomes now. Okay, all with the purpose to increase variation within the offspring that will, if they get this particular cell that has this recombinant chromosome, then you'll have variation in their genetics. Okay? Now, don't worry about the letters for now. This is just representing gene locations, the loci, and the actual genes. So let's say this is a gene letter C and whatnot. And this is actually a big C here. As we go into chapter 14 and inheritance patterns, we're going to talk about how genes are represented usually by letters, and capital letters are dominant characteristics, and recessive characteristics are the recessive lowercase letters. So in this particular case, you have the father that has all capital letters. Again, this is a little just a mistake here. This is capital C. The mother has recessive variations of the genes. And whenever they cross information, okay, when they do that crossover, if you look now, you have the father having a certain recessive allele, okay, meaning that for this gene, he is now heterozygous, and so is the mother. Don't worry about that for now. We're going to go into that much more detail in chapter 14. So ultimately, this creates a recombination of DNA, recombinant chromosomes. Okay? That's the main thing that occurs from prophase I. And this is very important because it helps variation, as I said before. The areas in which the crossovers occurred is called the chiasmata. And you can have one or more of these regions, as I talked about here, right? So this is a chiasmata region, another region also. You don't necessarily have to have one. Okay? Prophase I. Metaphase I, you have a lining up at the equatorial plate, at the metaphase plate here. Now, what's different between mitosis metaphase is that here, the homologous pairs are side by side. Okay, the homologous pairs are side by side. In mitosis, all chromosomes, it doesn't matter if you're homologous or not, are going to align at the center individually. But here, they align in pairs. Okay, that's all metaphase I is about. Now, there is something within metaphase I that occurs that's important for variation, and it is called independent assortment, where the arrangement that is happening, within the chromosomes. And what I mean by this, let's say here in one homologous pair, you have paternal chromosomes looking at one pole, the maternal are looking at the other. And in another homologous pair, the father's chromosomes are looking downwards and downwards here. Now, what this is trying to say is that each homologous pair will arrange itself, or orient itself, if I can say, independently of any other homologous pair. Okay? That in itself will create variation. Again, I'll get into that later on. And then you get into anaphase. Anaphase I, similar to mitosis, is just pulling, right? What is being pulled though? In this particular case, you're getting now the homologous pair are being separated. Okay? The homologous pairs have now been separated to each extremes. Okay? And you get telophase I and cytokinesis I. In telophase I, very similar to mitosis, depending on the species and the cell type, you can get the mitotic spindle disintegrating, the nuclear envelope reforming, and even an uncoiling of DNA. Okay? Or you can just continue straight. Just to give you guys your information. Also, I wanted to say, if you see the notes here, this should be more than sufficient to understand meiosis. Okay. So as I mentioned before, anaphase, there's a separation where they're held by the kinetochores, and the microtubules are pulling to opposite ends. Telophase I, you're having this particular event occurring of the nuclear membrane and so forth. And then cytokinesis 1, where you have that separation of the cytoplasm. And then at this place, you have let me just go up, a haploid cell. So take note of this. At this point, you have a haploid cell forming. And there's two of them. So you have two haploid cells. These will then further undergo another cell division, which is meiosis 2. Now, visually, it looks like mitosis because now you just have the chromatids, okay, the sister chromatids. And prophase 2, you have the disappearance of the nuclear envelope, the formation of the mitotic spindle, and so forth. But here, now, you have an alignment at the center. So if you take a look, now there's only the X's, right? There's only the sister chromatids, all align, the metaphase 2 process, and then anaphase 2, where then you separate the sister chromatids to their chromosome form, right? So just each individual little stick there will go to opposite ends, and then you would get telophase 2, okay? What is important to note always is that you have haploid cells only. Each cell is haploid. And then these will undergo cytokinesis 2, where you have 4 resulting haploid cells. And then that's it. Meiosis 2 and meiosis in general has finished. Okay? Very important key point is that DNA does not get copied. Okay? No copying occurs. Okay? Make sure you understand that there's no copying, there's no interphase going on, right? So it keeps consistent with the haploid number. Also, as some of my students tend to get confused with, do not consider counting each individual chromosome as haploid and then like a super half of that number of chromatids, right? At this point, you have just the copies of each DNA. So the chromatids, the chromosome and its copy, are still containing the same genetic information. So there's no reduction of genetic information because it's a copy. Okay, so understand the amount of genetic information, a set from the father and a set from the mother, as opposed to a chromosome and its copy, the chromatids, right? You'll still maintain the haploid number. Okay. So I want to talk a little bit more about the crossing over in detail. Now, if you're an AP student, you don't necessarily need to know this, but again, since we're going through everything of the Campbell Bio book, undergraduates might require this. Okay. So crossing over and that whole process of synapses, okay, is occurring during prophase 1. Now, what exactly is happening? Definitely prophase 1 is a very busy time. And here, in this particular instance, these diagrams you see here is a very nice visual of what is happening. So you have your homologous pair, looks more like this, right? So you have your maternal sister chromatids. It's quite difficult to see here, but since they're right now little strands, here's one little strand and another strand. Okay, and the father's chromosomes also. So you have the homologous pairs side by side. The chromatids are held together. Okay, each little red line that you see here and each little blue line that you see here are the chromatids that are paired up. Okay, that are side to side. And then these homologous pairs will basically align themselves precisely at each particular loci of the genes, okay? Because of the cohesion proteins, they keep everything stabilized. And also, there's the formation of what is called a synaptonemal complex. And this is a set of protein structures that will enable the homologous chromosomes to stay in their place very well. Okay, so you can see this occurring here. I apologize for the kind of grittiness of the picture here, not Casco quality. So ultimately, you have this complex that will kind of anchor both homologous chromosomes side by side. And then at very specific areas, you will get a clipping of the DNA. So I'm just going to go a little bit higher here. So if you look closely, there's a little nip of the DNA of one of the sister chromatids, and then here's another one, a little clipping there also. And the same thing occurs here and here. Remember, just one of the chromatids from each of the homologous pair. And then, as we go along, those clipped areas will then get exchanged. So they'll connect with the other and create what you'd see here. Okay, this little diagram that you see here. So now the maternal will then join with the paternal chromatid and vice versa. And ultimately, what you would get is a recombination. So now you have the paternal chromatids having a piece of the maternal DNA and vice versa. These locations in which this event occurred is called the chiasmata, okay? Or chiasma for singular. All of that is happening in prophase I. Prophase I. Okay. And that's pretty much it in terms of the meiotic processes. Comparing mitosis and meiosis, I strongly recommend understanding this table. It's like a keystone question in tests and exams and whatnot. You know, compare and contrast mitosis and meiosis. That's like a staple question in tests for education. Meiosis halves the total number of chromosomes, so it's a reduction cell division where you produce haploid cells. Mitosis conserves the number of chromosome sets. Meiosis produces 4 cells that are different genetically from the parents. Mitosis produces identical cells, even at the genetic level. Okay, and you can see this little visual here. I strongly recommend for you to study this, you know, to really understand and try to explain it to yourself so you can understand what is happening. Aside from that, prophase, you have the homologous pairing, and in mitosis, you do not have homologous pairing. Okay, and of course, meiosis, you get an additional cell division. Okay, so take a look at the summary table that I'm showing you here. Has a good amount of detail for you to study. All right. In terms of unique events, I'm just gonna go quickly over these since I just explained this right now. Synapses and crossing over. Of course, you have this area where DNA content is being passed through each of the non-sister chromatids. Okay. Homologs at the metaphase plate, where you have that pairing of the homologous chromosomes, and they're independently assorted amongst other homologous chromosomes. Okay. Of course, and then the separation of the homologs, so you get different recombination of DNA, of genetic information. Okay, I will continue here this scientific skills exercise for you guys to take a look at and try it out on yourself. Okay, in the concept check, that's always good to review these questions here. All right. And lastly, this could be given as an essay question. How do you see genetic variation within this process? Okay. As we go along, and we're going to look at mutations more, mutations in themselves are the key source of genetic diversity. What is a mutation? It's a permanent change in the organism's DNA sequence. A base pair could change, a whole chunk of DNA could be copied into another area, and so forth. So because of these mutations, you can get different variations of genes therefore a characteristic or a trait, and this is referred to as an allele, a different form of these genes. Okay, and then once you have these variations occurring over time and generations as organisms interbreed with each other, you start to get a shuffling of a variant from the original gene, and you start to get what is called a heterozygous organism, right? Again, there's something we're going to see later on. Because there's a reshuffling, that helps sexual reproduction, and that helps offspring have a unique set of traits there. Aside from that, you get other ways in which you can enhance this variability, being made from the mutations. And so you get three mechanisms: independent assortment, you have crossing over, and you have random fertilization. So independent assortment, which what I mentioned before, is that you get during metaphase, right, a different orientation amongst the homologous pair. Take a look at possibility 1, where all the paternal and maternal chromosomes are facing a particular side. Or you can just get, out of pure chance, possibility 2, where you get one homologous pair in which the colors are one way, and then another homologous pair which the colors are the opposite way. And because of that, when you undergo meiosis 1 and 2, ultimately you're going to get a different set of chromosomes. In this particular case, this combination number 3, combination number 4, and 2 and 1 and whatnot. And then these 4 cells, resulting cells, sorry about that little picture there, I was kind of emphasizing that these particular cells will then later develop into, for example, sperm cells, okay, and whatnot. It's a little bit more, the mechanism of the ovum of egg cells is a little bit different. You get polar bodies, and then you get the actual eggs. So there's a lot of some subsequent processes that occurs. But for simplicity's sake now, I'm just going to talk about sperm cell. Okay, so you get independent assortment. Ultimately, what does that mean? Just to give you like a formal definition there, that each homologous pair segregates independently over every other homologous pair during metaphase 1. Okay, so you can get a big combination of possible combinations of chromosomes. Okay, the book states that humans have 23 pairs, so since you need 2 pairings of chromosomes, 2 to the power of 23, about 8.4 million possible combinations. Having that said, then you have another layer of variation for the possible offspring, which is crossing over. Crossing over itself, as I mentioned before, you have DNA swapping. The resulting chromosome is referred to as a recombinant chromosome because you're just combining genes from each parent. And so you have now the offspring that will have this kind of recombinant chromosome, aside from the orientation of the homologous chromosomes, adding another layer of genetic variability. The number of crossovers does vary between the chromosome pair, right? The size of the chromosome, the position of the centromeres can enable more than one crossover to occur. As you can see here, you can have crossing over occurring in 2 different locations. If the chromosome is big enough, you can have this swap more. Of course, as it's smaller and smaller and smaller, it's a lot more difficult for that to happen. Okay. And lastly, random fertilization. So this adds that third layer of variation, right? Because any of those 4 sperm cells, any of those 4 sex cells can, be, I don't want to say selected, but by chance, one of those 4 will fertilize one of the eggs that are being formed. And then that in itself adds randomness and possibilities, right? So after all of these processes being combined, right, you get in the trillions in terms of possible combinations. So that is quite mind-blowing how we in the end, are all unique from each other, right? So evolutionary adaptation depends on population's genetic variation. The more varied you are as a population in terms of genetics, the more likely you can have the traits to survive and continue the species. A population evolves through differential reproductive success of its variant members. Okay? Each individual that is best suited for the local environment will leave the most offspring because they have the better traits. And if they have the better traits, they will pass down those traits to the offspring, which will then produce their own offspring, and so forth and so forth. And then that's ultimately the idea of surviving natural selection, adapting, and evolving, okay, at a more macro scale. We're going to see this later on also. So, but, this depends on the environment. And if the environment changes, maybe those organisms that have been lucky and reproducing, if the environment changed so much, maybe they're not the ones that have the best traits anymore, right? So it does depend on the environment, okay? So mutations are the original source of different alleles, and then there's a mix and matching during meiosis. So that's quite interesting. Mutations is king. So new and different combinations of allele can work, better than what was previously prevailing. So it all depends on the environment. Interestingly, though, when the environment is stable, many organisms that can do both forms of reproduction, asexual and sexual reproduction, can switch, right, to asexual reproduction. Because if the environment is stable, you want to produce more and more and more of your population so you can dominate that particular area in periods of calm, if I can say. And when there's more instability, you want to have sexual reproduction just for the sake of getting variation to ensure that they can survive most likely, okay? Sexual reproduction in itself is quite expensive in terms of, that's interesting there, it's quite expensive in terms of doing, right, of energy costs, of the whole process of recombination of DNA, of mating and finding a mate, right? All of that is energy costly. But in spite of that, sexual reproduction has, ultimately been shown to be across, a wide variety of life, and it is the driving force that enables us to survive in the environment. Okay. So while a few species are capable of reproducing asexually under unusual circumstances, animals that always reproduce asexually are actually rare. One example of this is the beloid rotifers, and this is a group of species that has, lived in a variety of environments around the world, and they reproduce only asexually and haven't engaged, supposedly. I don't want to say supposedly. Doctors, scientists have, analyzed this. Haven't engaged in sexual reproduction for 40 million years since their evolutionary origins. So that's quite interesting there. And then the book kind of discusses, does the group, showing these properties, is it the exception that proves the rule, or is it the exception rather than the rule? Okay. But, setting that aside, sexual reproduction and meiosis is the process that will drive variation in the organism or in the population. Yeah, with that, we finished chapter 13, section 3 and 4. And just to show you here the chapter review for you guys to have. Here's the first clipping, the first page there for the chapter review. Okay, you guys can pause to see that. Let me zoom in a little bit. Yeah, I'll give you some time if you need to pause there. And then the second page. Let me get this out of the way here. Oh, I'll pan down. Okay. And Here you go for that. All right. Great. So thank you very much for watching. I hope this video was helpful, and I'll see you in another one.